Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore absorptions and action spectra. At the end of this lesson, you will be able to interpret absorption spectra of chloroplast pigments and action spectra for photosynthesis. All right, let's get started. Absorption spectrum. So what is absorption spectrum? The absorption spectrum is a graph that shows the amount of light absorbed by a pigment at each wavelength of visible light. So basically, we want to study how much light is absorbed by a particular pigment, for example, chlorophyll A, okay, from the wavelength, for example, 400 nanometer all the way to 700 nanometer. So this spectrum indicates which wavelength of the light are absorbed most strongly by a particular pigment, and commonly measured for chlorophyll A, chlorophyll B, and some other accessory pigments, for example, carotenoids. So therefore, it is very useful to have a purified pigment by using column chromatography, which I have discussed about this in the previous extended lesson. So this is an example of absorption spectra consisting chlorophyll A, B, and carotenoids. So in this absorption spectra, we can see that chlorophyll A absorbs most strongly in the blue, about 430 nanometer, and reds about 660 nanometer region. Chlorophyll B peak in the blue regions about 450 nanometer, and orange red regions about 640 nanometer region. How about carotenoids? Carotenoids absorb light mainly in the violet to blue-green regions, about 400 to 500 nanometer, and some carotenoids can absorb up to 550 nanometer wavelength. However, you can see that the light in the green region, 500 to 550 nanometer up to 600, is absorbed very poorly by the photosynthetic pigment. So instead of being absorbed, the green light is mostly reflected, and this reflection of the lights leave their characteristic green color. So the question here is how we can produce this absorption spectrum. So in order to produce absorption spectrum, we need to use an equipment or device known as spectrophotometer. So the absorption of the light by pigments at different wavelengths can be analyzed using a device known as spectrophotometer. So in this diagram, you can see that this is the pigment in a special container known as cuvette. So we can place it inside this into this chamber. So the light will be emitted and pass through this pigment. So the pigment will absorb some light intensity and leave some other light to pass through. So when the light pass through, we can always detect it. So the principle of spectrophotometer, in this diagram, you can see that in the spectrophotometer, there is a prism to refract the light so that we can get a different wavelength. And then we have this, it has this filter to allow a particular wavelength of light to pass through. So for example, in this case, we allow the green light to pass through the chlorophyll solution. So some of this light will be absorbed and some will be transmitted. And we have these photoelectric tubes to show how much, to detect how much light energy or light is transmitted. From there, we can actually calculate, for example, in this case, because of the green light, okay, and we know that chlorophyll solution or chlorophyll A, B molecule, they have a very low absorption at the green light. So therefore, most of this green light can pass through and detected by the photoelectric tubes here and the galvanometer here shows a very high transmission. So a very high transmission indicates low absorption. On the other hand, if we allowed the blue lights to pass through this chlorophyll, and we know that chlorophyll molecules, they have a high absorption at blue and red regions, right? So for blue lights, they absorb the most here, allowed a small amount of light to pass through. So therefore, the galvanometer here show a very low transmission, which indicates high absorption. So as a summary, we know that white light can be split into its component wavelength, okay, by using a prism. And each wavelength can pass through a sample. For example, in this case, chlorophyll solution. So we can always start with 400 nanometer wavelength, 410, 420, and so on, all the way to 700. And each time we can plot the absorptions, okay? So transmitted light hits the photoelectric tube in this case, which convert the light energy into the electrical energy. And the resulting electric current is measured by this galvanometer, indicating how much light passed through. But for us, we want to know the absorption. So therefore, we can calculate the light absorbed by the pigment by using the value indicated by galvanometer here. For example, we assume that the light 100% arrive at the chlorophyll solution, and at the end, galvanometer show 98% of transmission. So basically 2% of light is absorbed. So next, let us look at the action spectrum. So the action spectrum is a graph that shows the rates of photosynthesis at different wavelengths of light. So this is an example of an action spectrum. So basically, this graph indicates which wavelength of light are most effective in driving photosynthesis. So we can have a plant, okay, in to carry out photosynthesis. We can measure the rate of photosynthesis, okay, by measuring how much O2 release per unit time or how much CO2 used up per unit time. So we can shine the plants, okay, subject the plant at different wavelength. We can start with the 400 all the way to 700. So at each wavelength, we can calculate the rate of photosynthesis and plot this action spectrum. So the spectrum is typically measured by the volume of oxygen produced per unit time or volume of carbon dioxide uptake per unit time at each wavelength. So we start with 400, then we get the rate of photosynthesis by measuring the volume of oxygen produced per unit time, and then we continue, repeat the experiment with 405, 410, and so on, all the way to 700, okay, nanometer wavelength. Okay, so the curve usually will have two peaks, one at the blue region, about 430 to 470, okay, at blue region, as well as one in the red region, about 660 to 680 nanometer wavelength. And there is a groove of dip in the green region, around 500 to 600 nanometer, because we know that green light is mostly reflected by chlorophyll, which are the main pigment in the leaf. So let us compare between absorption and action spectrum. So by comparing these two, you can see that both of them, the shape are quite similar. They match each other. So in terms of similarities, first of all, you can see that both spectra show peak in the blue and the red regions, okay? So about 400 to 500 nanometer, as well as 600 to 700 nanometer. And this indicates that the light in this region is most effective for photosynthesis. So the lights that absorb at this region to drive the photosynthesis, okay? So this is very important. You can see that in terms of the shape matches, okay? First, you can see that 400 to 500, high absorption, therefore high rate of photosynthesis. So basically, we can conclude that the energy the light energy absorbed by chlorophyll A, chlorophyll B, carotenoids, okay, is used for the photosynthesis. No question in this case. However, if you look at 500 to 600 nanometer wavelength in terms of absorption, very low, near to zero. However, in the action spectrum, the rate of photosynthesis is not near to zero. So the question here, why? So before we study or explain why the rate of the photosynthesis in the action spectrum does not show near to zero rate at 500 to 600 nanometer wavelength, when we see this absorption spectrum, that's near to zero light being absorbed. So let's look at the differences between absorption spectrum and action spectrum. So what it represents here. Absorption spectrum shows how much light absorbed by individual photosynthetic pigment. Action spectrum shows how the light is used, or which wavelength of the light is effective to be used in photosynthesis. In terms of curve profile, absorption spectrum show multiple distinct curves. For example, in this case, for chlorophyll A, B, and carotenoids, action spectrum shows a single composite curve represent the overall photosynthetic activity. In terms of pigment specificity, yes, for absorption spectrum, it shows the absorption characteristic of specific pigment, for example, pigment chlorophyll A, chlorophyll B, as well as carotenoids. For action spectrum, no, the spectrum actually reflects the total contribution of all pigment to photosynthesis. Effectiveness of the light in the green-yellow region, about 500 to 600 nanometer. So absorption is minimal, near to zero, particularly chlorophyll molecules show no or very low absorption at the green, yellow, or green region. However, for action spectrum, photosynthetic rate is low but not zero because there are other accessory pigments that contribute to the energy transfer. Still remember, in this experiment, we focus on carotenoids, chlorophyll A and chlorophyll B. But there are some other pigment which has a very low concentration in the plant leaf, which act as the accessory pigment to absorb the light at 500 to 600 nanometer wavelength, and they transfer this energy to the reaction center. So this explains why the absorption spectrum show a very minimum, near to zero absorption of light energy, but action spectrum shows that the rate of photosynthesis is low, but not near to zero. In terms of the spectral breadth, or how wide the spectrum is, you can see that absorption spectrum show narrower shape in this case, because limited to the absorption peak of individual pigment. So each individual pigment will show a narrow spectrum, okay, compared to the action spectrum, because action spectrum shows the integrated effect of multiple pigment working together. Okay, so for example, if you go back to the action spectrum and also absorption spectrum, you can see that, okay, the contribution to the action spectrum by this absorption spectrum. So there is a combination of different type of pigment work together to give the final effect, okay, on photosynthesis, which is translated to the action spectrum. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.